Hello, everyone. It is a pleasure to welcome you to this session and to this conference in its new format. So this year we have made some significant changes in respect to the conference. It is now structured around 9 priority themes aligned with the Latvia 2050 strategy. And in comparison to the previous years, it is more focused, and it is in a 3-day format. So this is the first time we're trying it, and we will be happy to receive any feedback on what you think about it and how we should do it the next years. Don't be shy. In respect to organizing the conference, I guess I should have started by introducing myself. I'm Antra Boča, head of the research office of the University of Latvia. And so the research office was in charge of organizing the conference, and hence I will be the first to admit that we might have not done the best job in terms of communicating about the changes in regards to this conference. And basically it was a lack of time and lack of capacity, and I'm not trying to make up excuses, but it's just the reality. However, it is also exactly why this moment matters, because this conference is not the end of a process, it is the beginning of a process. And this year, we are starting to work on the university strategy and including our research priorities. And we don't want to do it behind closed doors, but we want to do it together with the researchers. And because who knows science better than scientists themselves? And I would like to use this moment to invite everyone to engage, question, and help shape what comes next, including in terms of how we will organize the conference next year. So I would like to invite you all to ask yourselves, what will matter in your field 5 years from now? What are the biggest challenges we should work on, and how should we approach them? This theme is about environmental sustainability and climate change. So let me briefly put this into a broader context. Yesterday at the plenary session on global competitiveness, we heard that the next EU planning period, there will be around 700 billion euros assigned to climate policy. There's a clear goal. The goal is to reduce the dependence on fossil fuels and move towards sustainability. And we, of course, in Latvia don't have fossil fuels, but we are rich in natural renewable resources and knowledge surrounding it. And I think the current situation presents an opportunity, not just to adapt, but to lead in some fields. But for someone to be able to lead the person, us, we need to have a vision. And as scientists, our vision should be bold. And today's session speaks directly to this opportunity. We are living in a world where fossil resources are decreasing, natural ecosystems are under pressure, and mountains of waste continue to grow. The traditional view sees waste only as a problem. But many scientists, including myself, we see waste also as a resource waiting to be unlocked. When I was studying environmental sciences at the University of Latvia, we were told that plastic can be degraded only by UV light, and that's about it. But today, we will hear about microorganisms capable of degrading waste, meaning organisms living in waste environments with the potential to turn one of our biggest challenges into a potential resource. And it is not only about microorganisms making something disappear, it is the power of enzymes allowing to deconstruct plastic into its raw form to be reused again. And think about it, this is crazy. We see Getlini growing and growing, but maybe one day that is where we will have mine the next resources. At the opening plenary discussion, Professor Ambainis made an interesting point. In his field, of computer science. He currently sees how ideas developed by him and his colleagues 20 years ago are hitting the market, basically, in the form of products. And he said something worth repeating. The general role of scientists at universities is not to develop products that can hit the market in 3 years, because that's something the software companies will do for you, but it is to create knowledge that will change the world in 20 years. The impact of science is long-term. And at the opening session, we also heard an interesting example by Professor Umea from Tartu University of how a practical problem led to an idea to fundamentally rethink geospatial visualization, from flattening geospatial data to working with spherical structured data. And what stood out to me was not just the idea itself, but the mindset behind it. The willingness to question assumptions, to revisit forgotten concepts, and to make the argument that this is the right time to change the status quo. And I think that many researchers also in this room have great ideas, and the only barrier to truly engaging with them is not the lack of knowledge, but the lack of confidence. And confidence grows through discussion, through critique, through refining ideas together. And this is exactly what this conference is about, and what this conference should ignite: the willingness and interest to discuss ideas that will lead to impact. And then turning to the second, so we will hear this one talk today about microorganisms, and then our second talk will be about sustainability and the university's roadmap towards it. And regarding this, I was reminded of something that I heard a lot during my PhD studies from my colleagues that were doing their studies in wildlife management and environmental management. They like to say, nature does not to be managed, people do. And so I'm happy to see that in this theme of environmental sustainability and climate change, there were sections that linked environmental science and sociology, and even humanities. Because what drives us often are the aspects that are not rational. It's our feelings, it's our views, our values. And I hope this interaction grows more and more. Because we cannot manage nature in the way we often imagine, but we can and must manage human behavior, systems, and institutions in a way that aligns with natural limits. And universities have a special role here, not only in generating knowledge, but in shaping values, practices, and future leaders. So let me close with 2 invitations. First, today at 5 p.m. on the second floor, there will be our closing session, where the Vice Rector for Research will say some final words and also give some numbers regarding the conference and the process. And we will also be glad to hear feedback. So everyone's welcome, 5 p.m., second floor today. There will be pizza. Please come, because otherwise, we have ordered a lot of pizza and someone has to eat it. And second, so let's take what we have heard during this conference and we'll hear during this conference to think big, and to think of our vision for the future. Let's challenge each other. Let's discuss ideas that may seem too bold, too early, or a little bit too crazy, because only such ideas drive real change. And thank you, and I wish you an inspiring session. And I want to introduce the next speaker, Professor Olga Mutter, who will give a talk on evaluation of functional capacity and plastic degrading potential of Bacillus spp and other bacteria derived from Gettlinge landfill. Thank you. The floor is yours.
Yeah. Dear colleagues, dear organizing committee, it's my great pleasure to present our results at the university conference. And I would like to introduce you with our main results on plastic biodegradation, which have been obtained in frames of our 3 years project financed by the Latvian Council of Sciences and the University of Latvia and Latvian Biomedical Research and Studies Center were the main partners. Also, Getlini Eco Limited has been actively involved, and also students from both partner institutions, as well as 2 foreign master students, Sara Pereira from Portugal and Luisa Gartling from Germany, contributed this work a lot. We just finalized this project in December 2025. 2 weeks ago, our final report has been successfully approved. So the topic is really huge and global. Plastic pollution is an ever-escalating issue with detrimental effects on both the environment and human health. This presentation embraces not only our data, but also global trends in plastic waste management, such as the waste flow system, the main aspects and key discoveries in plastic biodegradation, and others. Plastic production has sharply increased over the last 70 years. In 1950, the world produced just 2 million plastics. Now it produces over 450 million tons. And plastic has added much value to our lives. It's cheap, versatile. However, when plastic waste is mismanaged, not recycled, incinerated, or kept in sealed landfills, it becomes an environmental pollutant. And plastic pollution is mostly about how waste is managed, not how much is produced. How waste producers do not leak much waste into the environment, their pollution is close to zero. In turn, when waste is not collected, safely disposed of, or properly treated, it becomes a pollutant and leaks into the environment. Plastic waste accumulation represents a persistent environmental challenge driven by rapidly increasing polymer production, short product life cycles, and insufficient end-of-life management. Despite substantial technological progress and increasing policy attention, effective plastic waste management remains constrained by a combination of technological, economic, and policy-related barriers that limit the scalability of circular plastic systems. And these barriers are highly interconnected. Overall, the classification of plastic polymers is vital for understanding their diverse properties and recyclability. And polyethylene, both high density and low density, as well as polyethylene terephthalate, are the most widely produced plastics. Effective plastic waste management requires the integration of degradation mechanisms with control, pre-treatment, chemical conversion, or energy recovery processes guided by environmental safeguards and circular economic priorities. A conceptual scheme is presented in this diagram to illustrate the major plastic degradation and conversion pathways and their integration into circular resource recovery systems, summarizing photo, thermo, biodegradation, mechanical recycling, chemical depolymerization, and thermochemical conversion routes leading to recovered materials, fuels, chemical, and biomass within circular plastic systems. Among findings in plastic biodegradation, which have significantly advanced the potential to convert plastic waste into safe, reusable monomers, the key discoveries are as follows. Causes so-called plastic-eating bacteria. For example, this famous Adenella sacchaensis discovered in 2016 at a Japanese recycling plant. Engineered super enzymes, fast PETAs, plastic-degrading fungi, insects and larvae, and plastisphere, or in other words, biofilms, and enzyme-embedded plastic. The studies on plastic biodegradation embrace the mechanisms, microbes and enzymes, biodegradable plastics, factors affecting biodegradation. And many conventional plastics remain in the environment for hundreds of years, even up to thousands years, as it is predicted, before breaking down into or sometimes bypassing the microplastic stage. Biodegradation of plastic is an eco-friendly process where microorganisms break down plastic polymers into water, carbon dioxide, and methane under anaerobic conditions. And it involves microbial colonization, secretion of enzymes to break down polymers into monomers, and finally mineralization into harmless product, often within 3-6 months in suitable conditions. This optimistically. And the limitation is that biodegradation is generally slow, making it difficult to use as a primary industrial large-scale biotechnology solution at present. The biological degradation of plastic generally involves breaking down long polymers into short oligomers, dimers, and monomers, and extracellular enzymes, for example, depolymerase, hydrolase enzymes, induce hydrolytic cleavage in polymers. In addition, free radical formation can also cause a change in the number of bonds between monomers. The action of hydrolytic enzymes such as depolymerase, oxidase, and peroxidase causes an increase in the hydrophilicity of the polymers, thus increasing its rate of biodeterioration. Landfilling and incineration are the most established method for plastic disposal. Landfills act as significant long-term sources of microplastics and chemical byproducts resulting from the partial biodegradation and fragmentation of plastic waste, including microplastic and bioplastic. So, the microplastic removal efficiency upon leachate treatment remains challenging and varied from 3% up to 100% in dependence on technologies used. Biological, physical, chemical processes can be applied upon leachate treatment. Among biological methods, aerobic and anaerobic, lagooning, activated sludge process, trickling, filter, moving bed biofilm reactors, and other approaches are discussed and developed. A study on microplastics distribution dynamics and the plastic biodegradation potential in landfills is still limited, and landfilling can promote the formation of microplastics in landfill systems, and landfill depth is significantly correlated with the relative abundance of microplastic sizes, generally with smaller microplastics becoming more prevalent at greater depths. About bioplastics. Bioplastics derived from renewable resources like corn or algae are increasingly used to reduce fossil fuel reliance, primarily in packaging, agriculture as mulch film, and consumer goods. And importantly, only 36% of bioplastics are bio-based but non-biodegradable. And the efficiency of bioplastic degradation depends on polymer complexity, crystallinity, molecular weight, as well as environmental conditions. So therefore, further research on the factor affecting the biodegradability of bioplastics, their different types, is necessary. Proper end-of-life treatments, such as industrial composting and anaerobic digestion, are crucial for realizing biodegradation benefits. And importantly, also bio-based, biodegradable plastics have a limited impact on overall energy demand. They consume substantial water for biomass production. So future research should address economic and policy challenges in this area. And the second part of my presentation is focused on our data, on our results within this project on biodegradation of plastic. So, since landfills are enriched in plastics, microbial dark matter can hide potent plastic degraders. Uncovering of novel plastic degrading microorganisms by exploring natural habitats not only can directly provide effective tools for processing of plastic, but also serve as a template for modeling artificial systems to combat plastic pollution. The goal of the project was exploration of microbial communities residing in municipal solid waste landfill to assess their potential to degrade plastics within the landfill ecosystem. Gatlini landfill is managed by SIA Getlin Eco, and the landfill area is 87 hectares, and it currently receives up to 1,000 tons of waste daily. Our recent paper summarized different aspects of plastic microbial degradation, and I will start with collection of samples and metadata, which has been performed and summarized by our colleagues from the Latvian Biomedical Research and Studies Center. So the sampling site was situated on one side of the old dump hill, which was active from the early 90s. Around the year 2000, the landfill underwent significant modernization and the old dump site was closed. Therefore, the collected solid waste samples were at least 20 years old. The collection sample was supervised by expert from Getlin Eco Limited to ensure safety of personnel and the environment. For collection of solid waste sample at range 1 meter wide approximately and 4 meter deep and stretching around 18 meters in the waste hill was dug with a mechanical digger. Mixed waste samples were collected in containers driven into the walls of the trench at 5 different distances from 8 meters to 17 meters and different depths from 1 meter up to 4 meters. The landfill microbial communities are dominated by bacteria, 21% of reeds, while archaea are 20 times less abundant. Also, bacteria were also the most diverse, spanning 50 phyla. Actinomycetes were the most abundant. Archaea, dominated by Eurarchaeota, included 101 reed. Bacteria in soil fraction samples were represented by approximately 1,700 genera. Among these, the most abundant were Streptomyces, Pseudomonas, and Sulfuricaulis. Among fungal genera, representatives of the phylum Ascomycota were the most abundant. To characterize the microbial communities residing in the municipal waste landfill, a total of 30 soil and leachate samples were selected for sequencing analysis. In general, with the highest relative abundances were Sulfuricaulis, the Archaea Metanotrix, and bacteria Pseudomonas. Functional processes associated with landfill microbial communities confirm processes such as hydrocarbon degradation, methyllotrophy, methanotrophy, methanogenesis, sulfur oxidation, and other functions, and analysis also indicated plastic degradation as one of these processes. To further investigate the abundance of microorganisms, plastic degraders, the Plastic DB's database was searched for records on detected species. The microbial communities from the soil fraction samples collected from the old dump hill contained 31 potential plastic degrader. And the search for potential plastic degrading enzymes revealed 200 candidates in the landfill microbiome, including commonly known enzymes such as hydrolysis, PETases, and lacases. The enzyme profile was different from that found in agricultural soil, which was relatively free of plastic. And the top 20 plastic degrading enzymes identified in landfill soil based on plastic DB resources are shown in this diagram. Enzyme names include enzyme type and taxon from which the enzyme was first derived. And after all this huge metagenome of Getlin landfill has been tested and summarized, so we started a laboratory modeling experiment with landfill samples. The first study presented today was aimed at assessing biodegradability of bioplastics in the presence of landfill leachate. The idea of this study was proposed by Katrina Sipola, a pupil from the middle school French lycée in Riga. She came to our lab to work out the scientific research project, and Katrina brought cassava-derived biodegradable plastic plates and package materials from Café M, which was the first zero-waste coffee shop in Latvia. And we decided to expand this study with implementing landfill leachate, metagenomic analysis, microbiological, biochemical, and other methods. Biodegradation of cassava-derived bioplastic after the 30-day incubation in a 2% leachate was higher in the set with agitation, comparing with other sets, not agitated leachate set and soils. And interestingly, the regression analysis showed a high correlation between weight loss and reducing of a surface area during degradation process. Finally, it was concluded that among bacteria genera, which are known as potent bioplastic degraders, the most abundant were Pseudomonas and Bacillus. Landfill leachate in 2% concentration were shown as an appropriate environment for biodegradation of bioplastics. And this fact can be further discussed in two aspects. The first one, leachate as a result of a previous bioplastic decomposition upon waste treatment. And the second one, leachate as a source of microorganisms and respective gene families responsible for biodegradation of bioplastics under optimized conditions. The next study aimed to isolate, identify, and characterize bacteria with plastic degrading potential derived from the Getlini eco landfill, and to assess the degradation activity of a microbial consortium toward untreated and pre-treated low-density, high-density polyethylene, as well as polyethylene terephthalate. And later, our colleague Sara Pereira has successfully defended her master thesis on this topic at the University of Lisbon. In total, 47 microbial colonies obtained from different media and sampling sources, and among the bacteria selected, Bacillus was the predominant genus identified, and the screening of non-specific esterase activity was done. Following a 6-week batch experiment, a newly developed bacterial consortium consisted of 23 isolates, biological reduced the weight of untreated low-density polyethylene by 19%. Finally, it was concluded that among bacterial isolates, Bacillus was the most predominant genus identified, while metagenomic analysis of landfill leachate showed a prevalence of the Pseudomonas genus. Another finding is that addition of nutrients to the minimal medium with plastic powder as a sole carbon source resulted in a broader spectrum of isolates with target properties. A newly developed consortium derived from Getlini landfill demonstrated relatively high degrading activity toward low-density polyethylene, resulting in a weight loss of untreated microplastic granules up to 19% after 42 days incubation. And thermal, acidic, and basic pretreatments of plastic granules led to distinct differences in bacterial physiological responses. The novelty of the SORT study is related to the application of original enrichment protocols for biofilm development on virgin PET surfaces, as well as combination of a high throughput sequences and fluorescence in situ hybridization to reveal differences in microbial community structure with emphasis on abundance and spatial distribution of Pseudomonas in biofilm. This study was performed in collaboration with Professor Nuno Felipe Azevedo and his research team from the Faculty of Engineering, University of Porto, Portugal. Our colleague colleague, PhD student Laura Jorge, has visited Portugal to bring samples and take part in visualization of these biofilms. Different nutrient composition upon laboratory enrichment resulted in different activities of bacteria immobilized on PET surface, and these data show the differences in fluorescein diacetate hydrolysis activity and microbial community structure in biofilm developed under laboratory model conditions. Also, we tested the biofilm from PET bottles, which were weathered under real landfill conditions at least for 20 years. FISH method was carried out to identify the presence and distribution of Pseudomonas genus bacteria in biofilm on plastic surfaces, and so yellow color attributes to Pseudomonas, and green color non-Pseudomonas. So it was suggested that the environmental conditions associated with landfill sites, such as exposure to diverse microbial communities and fluctuating physicochemical parameters, may enhance the colonization and proliferation of Pseudomonas on plastic surfaces. And the next study was focused on the anaerobic digestion process of 5 cellulose-derived polymer types: cellulose, cellulose acetate, acetate textile, viscose, nanocellulose, and for comparison, heteropolysaccharide, pectin. The fate of cellulose-derived polymers during the anaerobic digestion of organic wastes differs greatly and has been poorly explored. The most important factors influence the outcome of anaerobic digestion are the physicochemical properties of polymers and external factors such as temperature, oxygen, pH, microbial community structure, operation mode, retention time, pre-treatment, additives, inoculum acclimation, food microorganism ratio, and others. And the increased crystallinity and the high carbon to nitrogen ratio of the polymer decrease its biodegradability. This study has been initiated and supervised by our colleague, Dr. Andres Greenbergs. Unfortunately, he passed away. And he had a lot of original ideas how to continue the direction of anaerobic biodegradation of plastics. So, monitoring biogas production during fermentation revealed considerable differences among the samples. Specifically, cellulose produced the highest biogas volumes, which was followed by viscose and cellulose acetate. In turn, the control, without any amendments, nanocellulose and pectin did not produce biogas under the tested conditions. Metagenomic analysis revealed that the proportion of bacterial and archaeal domains in the landfill leachate samples after the 147 days fermentation varied greatly depending on the type of polymer added. Cellulose acetate and cellulose showed the highest relative abundance of archaea. In turn, acetate textile and nanocellulose had comparatively lower abundance of archaea and lower cumulative biogas production. At the phylum level, archaea were represented by Euryarchaeota with relative abundance up to 80%. The cellulitic activity was tested in the intact samples, as well as pellet and supernatant, which was obtained by centrifugation. Enzymatic activity in the supernatant is expected to reveal an abundance of extracellular enzymes. As shown in this figure, the cellulitic activity in the intact landfill leachate samples and the pellets and supernatants differed depending on the type of polymer added. Based on the results, we can conclude that landfill leachate is appropriate for the anaerobic biodegradation of cellulose-derived polymers. However, the process is polymer specific. At the same time, some of the results differ from what has been reported in the literature. Hence, the specific physicochemical characteristics of leachate should be considered. And finally, briefly about upcycling plastic after biodegradation, using enzymes or engineered microbes to break down plastic wastes into monomers, which are then converted into higher value products like specialized bioplastics, chemicals, or fuels. This approach transforms waste such as PET and polyethylene into sustainable materials, offering a bio-upcycling circular economy solution rather than mere disposal. And developing robust whole-cell microbial catalysts for plastic degradation is an important step toward addressing the environmental accumulation of hard-to-collect plastics, particularly microplastics, for which no alternative technologies exist. Primarily, most efforts to date have focused on engineered microorganisms that can function well under controlled laboratory conditions. These strategies are far from relevant operation settings, such as wastewater treatment plants, where the removal of microplastics from sludge is urgently needed, and variables like pH, temperature, salinity, and nutrient availability are difficult to control. Bridging the gap between lab-based proof of concept and real-world deployment will require not only innovative microbial engineering, but also careful integration with environmental, regulatory, and societal consideration. Thank you for your attention. I read my speech to make it in 30 minutes. Thank you very much. Olga, do we have questions?
Impressive work, so many days. What would be like kind of one to take? Oh, sorry. Hi. Yeah, so impressive work, a lot of data, a lot of research.
Thank you.
It's looking impressive, yeah, indeed. So, what would be kind of like 2-3 takeaways that you would kind of like to address to us, or maybe especially from the Gatling landfill, like what's kind of you would like to emphasize on? I mean, there's a lot, yeah, but
I think that this leachate, which is very variable and changing in its chemical composition, bring a huge resource for our further investigation, and we should use this, to elaborate some novel methods, technology,
Thank you for the presentation. And I was wondering, what is your vision of the future? Like, will Gatling be a place of resources for us in the future?
I think so, because Gatling are actively working and they have a lot of projects, research projects, where they optimize tunnel composting technology for treatment of organic wastes. So we collaborate with them, and I think that, and they are young, young people. This is very, very important. So I think that in future, the combination of technology should be applied, and as I mentioned in my presentation, combination of physical treatment, chemical pre-treatment, and some filtration and so on should be applied to get the result. But regarding upcycling, I think that we are at the beginning of this way because these laboratory findings are too far from practical application.
Yeah, but I think because it's the beginning, it's the best time to jump in. It's the best time to start here. That's, I think, something, because there are fields that are so crowded and there's no way we could compete, but something that's brand new, where there are not many people working on, that's where we should jump on.
I agree with you.
Thank you.
Thank you. It's really impressive what you've done. And the question is, you have concentrated to bacteria mostly, yes?
Yeah.
At the same time, we know that the fungi play say, paramount role at the biodegradation process, also for microplastics. So what's your estimate? So are they present, or the environmental conditions in the landfill do somehow prevent fungal activities? Have you seen them? And would you make some suggestions how to proceed or how to include also this part of the microbial world into the plastic biodegradation things? Thank you.
Thank you, Professor. This is a very important question, and the metagenomic analysis has revealed a very small abundance of fungi in leachate and soil. In landfill, it was about 0.05% of all community. So on the other hand, we know that fungi are a powerful tool for bioremediation, biodegradation process. During isolation with selective media, we have not obtained any fungi. So maybe we have to look into this efficient fungal isolates using other alternative medium compositions and continue our study. But this project is over. Your idea maybe would be necessary for the next projects, proposals. Thank you. Thank you very much.
Hey, thank you for the introductions. Thank you for the presentation. I think we will have a nice jump towards a more broader view about sustainability, and I like how we, the whole presentation. Thank you, Olga. I was looking at the glass bottles that were the plastic bottles that were on the tables there, and thinking about how really scalable sustainability is. And I apologize for those bottles being there in the name of the university, but let's look at the big picture, and I think I want to start with a quote from Charles Dudley Warner, who is a fantastic writer and an author of a book, My Summer in a Garden, and he said that mud pies gratify one of our first and best instincts. So as long as we are dirty, we are pure. Of course, talking about the fact that gardening does make your hands a bit dirty, and I advise you to, and I kindly ask of you to look at this presentation as an invitation to collaborate and give your feedback on how the university can enhance its development in terms of sustainability. And on that note, I am Renārs Kairis, Head of Sustainable Development for the university. I am also chairing the Eco Council, which is an advisory council for the leadership of the university. And let's look at some of the activities that we have done. And also I'll showcase some of the problems that we have encountered. But when we scale sustainability and when we try to create a roadmap towards it, which isn't really a goal either, it's a never-ending process. A good baseline is to have a strategy, and thankfully we also have a specific goal related to that, which is the number 5.1 in terms of green thinking and sustainable university environment and administrative support. And this is really where our journey started. And thus now we have also implemented a sustainable development policy. And I think it's time to rewrite it because it still states that it is a voluntary inclusion of social and economic environmental issues in our daily plans and life and management. And I think back in 2020, it's still really, or 2019, it really felt like it. But now it seems that it really is just a matter of decency and a matter of understanding the global processes. But nevertheless, our policy states principles, directions, and instruments on which and the premises of which the university is going to develop. And I think largely the most important to mention, of course, being research, as we are in a conference that is dedicated around excellence in research, and also the participation and openness for the university as an organization. So the directions of which we rely on for the university, also being the fact that as an organization we have to be sustainable, we have to look at our resources or spending or investments. We also have to prepare the next generation of researchers, the students, activists, experts in this field. But also looking towards innovations, collaboration with industry, policymakers, pitching the fact that we have to act on climate change and social issues, and the instruments, of course, being those said innovations, procurement policies, or circular economy-based solutions, etc., and also participation in numerous programs. And on this note, the university is the only university, I think, in the Baltics right now, which has the green flag status by the Eco Campus program. And I like this program a lot because it combines preschool institutions together with universities globally across all continents, and thousands of schools and universities are let's say interconnected in this program. And the work starts with, of course, registering and then, let's say, mapping out the issues that the university has. And I think I'm proud to say that largely this initiative was a bottom-up initiative from our researchers. And together with the students, this was a pitch to the management at that time, which was warmly welcomed. And now we can safely say that these are the 7 steps of the Eco Campus program. And now it is our 3rd year where we have participated, and our 2nd year where we have the green flag status, and the green flag also itself flying next to the House of Nature. And the way you can structure sustainability in an organization's governance is, of course, very I mean, broad, interesting, and there can be a million ways to do it. But this approach presumes the forming of an eco committee. Then, of course, also carrying out an environmental research. And by the way, our colleagues, next week is going to be that week where you receive the survey about our habits and sustainability literacy. After that, we try to find ways on what the main problems are. We try to link it with the curriculum of the studies, but also making an action plan with additional interventions that could educate and make the baseline of the university greener. Then we monitor and evaluate how that goes. We form a dialogue. Half of the members of this committee are students, and I think it's a really important voice to be listening to. And the Eco Charter is the policy that we made after the evaluations and evaluations of our students, staff, and partners. And I really specifically like this picture because this is a picture of our first green flag at the National Library of Latvia, where the university and the biggest university in Latvia received the flag together with 3 preschool preschool education institutions. And some may find it amusing, but I find it as a statement of really a full circular moment where we need to teach children, students, and their parents from preschool education to universities. And so this is, I think, a really nice testament to that. In terms of how the university looks on a broader scale, we are ranked first in Latvia, and from time to time also first in the Baltics by QS or Times Impact Rankings. Times Impact Rankings are suspiciously nice to us by granting us the top 100 global places in terms of SDG 16, which is peace, justice, and strong institutions. And we also mentioned, as I mentioned before, the Eco Campus program and also some local sustainability initiatives are those of whom we take part of. And we use this as a nice roadmap as well to kind of just structure the work around what is the global tendencies. And this is not the goal to be high in the rankings. This is just a matter of being interconnected with the global processes. Since the implementation of said Eco Council, and also now with my position being established, since 2022, we have had more than 80 sustainability activities, 21 seminars, presentations, 23 educational and communication campaigns. If someone is not really local here, then Talka or Cleanups is a fantastic initiative that combines social, let's say, social advocacy and practical work towards cleaning up the environment. We have had several infrastructure projects. We now have more than 35 cooperational partners. We also have not only the existing urban meadows next to the House of Nature, but 1,000 square meters more where scientists experiment with how urban meadows grow, where students go and use it as a living lab to research the plants and species. And this year's monitoring reported 96 plant species, 8 insect species, and 7 snail species. Not all of the snail species being very friendly towards or biotope. And moving to, I will move towards some pillars or activities from which we work, and of course the planning and policy development being one of them. As I said, the Eco Council is an advisory council for the management. We also have several different structures working towards this, and those being, of course, head of sustainable development, then the council, and also lately what we have done is completely integrated also social sustainability issues in a very horizontal manner. And now having diversity, equality, and inclusion committee, also a working group. and soon to be an action plan also for these issues that are interconnected, because our sustainability policy states that social justice issues are non-detachable from sustainability, and our new diversity, equality, and inclusion policy states that diversity, equality, and inclusion is also inseparable from climate change and those processes. So I'm proud to say that we look on this very broadly. And now about the progress. I feel a bit ashamed not to have more data since this is a scientific conference, and I probably have a lot of researchers eyeing this statistics and wondering where did those numbers come from. But of course, taking year 2019 as the baseline, pre-COVID, before there was any remote activities. Towards this strategy KPIs, sustainability policy, eco-council initiatives, and also initiatives that come from the university staff and students, we have managed to, largely due to savings in electricity and heating, reduced our climate impact. A bit of a mistake there. It's scope 2 actually, by 32%. Our electricity consumption annually is down by 22%. The heating being one of the most pricey in terms of climate impact, also down to by 36%. What I'm really proud of, before we started this initiative, we didn't have waste sorting, but now the share of unsorted waste is down by 45%, and the share of sorted waste up by 83%. And also the consumption of water, besides the fact that we have a community garden that uses this water, is still down by 34%. And now on the fact of now on the topic of collaboration and engagement, I think I want to look into the fact that we have had several activities that were really initiated by our students, and this is a picture from one of the jury's visits when we first received our green campus flag. Also an exhibition from Get Linjeko, so also a collaboration there. And this is, I think I will get more into it in the next slides. But also combining the collaboration with sustainability in academic and campus life. We have had several really impactful projects, one of them being the community garden, which outlived its first season last year. And we hosted several working groups and seminars about the history of gardening, about the history of this region, about the history of the development of the academic center, combining it with some sustainability literacy facts in order to get more information out about the topics of sustainable development. And the feedback has been great or good. The total number of participants last year in the garden was 75 people in 45 garden beds. And the survey was completed by 37 people, which were mainly the owners of those garden beds. So not all of them, but 37 responded of a short survey that stated that 57% feel like the garden improved their sense of belonging at the university. 87% of people view that urban meadows and the implementation of them is a really positive impact. And 57% of respondents said that it improved their sustainability and environmental literacy. And the next question, after the question about sustainability literacy, was actually, well, what did you learn? And the answers really surprised me. There were people who said, well, I learned a lot about bee colonies, or I learned a lot about the impact of urban meadows and the rainwater systems. Some mentioned social issues as resilience as a part of sustainable development. And this data for me is really interesting because this project largely covers a lot of UN sustainability goals. And through projects like this and also public program and public activities that we have done, the feedback has been great. And I like to see how, let's just say that it makes people a bit uncomfortable at first, the idea of having a garden or the idea of having sustainability activities in the garden. But it really is a good intervention in a time where it's really hard to grab people's attention or educate on these topics. So also as a part of research dissemination, the public program of the community garden hosted around 150 people towards the whole day, and we could disseminate research results also to people, society, and our community. So this, I feel like, is a matter of is a matter that could benefit from more exploration. And of course, that being one of the ways we communicate and raise awareness about these topics, we keep people informed through our email listings and our sustainability projects, policies. We also have the communications department represented in the Eco Council. And we really do see this as a key pillar into making these activities possible. So again, one picture from the event from the community gardens, also combining some plant awareness topics led by one of our researchers, and the vice rector is also in the circle. There's students in the circle. There's local people who live around the university also there. So yeah, numerous ways on which we can advance sustainable development. And lastly, I think I want to mention, of course, not sure if I can say bastion of sustainable development for the university, but surely one of the cornerstones, and it is the academic center development. And just recently we welcomed the European Development Bank here, and she was also, the representative was fairly surprised on the creative ways on which we try to engage the students and staff, and with largely important investments, we are advancing also a large-scale modernization program. Well, I will show you the visualizations after this, but we have indicated a lot of ways on which we can build solar energy systems, smart building management systems. Well, if you go to the second floor in this building, there's no even light switches for the auditoriums or cabinets because it's all controlled by the system. And of course, we have the Green University Initiative, and the project is expected to save 1,598 megawatt hours of energy annually, reduce the primary energy consumption by 39%, and improve the energy class of each building also. And also, this is a part where we involve community and where I advise researchers to join, because in order to achieve these numbers, we really do need to have a reliable and trusting community. And I added the picture of the community garden as kind of an intro towards what the campus is going to look like, and also as a statement to the fact that any initiative that came from a student can be implemented into the development of the academic center and the university. So this is it for me. If you have any questions, feel free to, and if you have any ideas on what to change or what to do differently, or any initiatives, then we are very open to collaborating.
Thanks. I have a question. What are the criteria for university to receive a green flag?
The criteria were the 7-step procedure. So we have to form an eco committee. We have to make a self-evaluation every year. In this self-evaluation, we receive data for mobility, also the lifestyle of our students and staff, and then we evaluate how sustainable they are. And also in this evaluation, we lay out all of the data, which is how much energy is used, how much energy is used on the heating, how much water do we use, how do we sort waste, what is the number of food waste. All of this data combined, which is a 70-page long document, also together with our action plan that we have made from the evaluation, is then reviewed by the committee of the State Nature Foundation, and after that we receive the verdict whether we have the flag or not. But the methodology is actually difficult, and if I may say that QS and the Times Higher Education rankings together do not even, let's say, create a system that is that in-depth, complicated, but also for a good reason.
Okay, thank you. I have two more questions. I don't know if I can ask them right now or do I switch with someone else? But there's another question for me. It's like, do you plan to collaborate with Botanical Garden regarding community gardens?
Funny enough, my colleague Renate, which is the director of the Botanical Garden, actually, I invited her to help me on the community garden here on campus because she had the initiative Sportapils Dārzi and made the biggest community project in Riga thus far with over like 100 or 200 people. And so I invited her and then she had she saw this possibility to apply to the Botanical Garden, and now after making this community garden, she is now working there. And yes, we have collaborated, and we are actually working towards a project on communicating plant blindness together with the University of Agder from Norway. So yes, that is very much in the makings.
Thank you. I'm very happy to hear that. And then another question, which is maybe a little bit controversial, is like, do you feel like the move towards eco-campus is more sustainable than using already existing building in the city center? That's regarding like the fact that university had a lot of different buildings across the entire city. Do you feel like building a mega campus is better than in sustainability wise than keeping those buildings?
A very fair question, and the baseline of let's say the development of a single campus was really first of all the problem of having tens of buildings scattered across the city, which made collaboration ineffective, and also the concentration of a science excellence center really ineffective. Also, those being the most energo-inefficient buildings, which do use a lot of power. The premise is also the fact that the Riga City Council has declared this land as land for development. That was prior also occupied by gardens that were kind of let down. So what we are doing is we are not occupying, let's say, biodiverse areas. We were kind of restoring the previously abandoned garden territory in this area. Which is also a controversial opinion, maybe, but at least in this part of the area in which the Riga City Council has declared as a zone for development, that is actively what we are doing. And also the campus itself, related to the vision of Sponge City, will implement the natural drainage systems, similarly to what meadows also do, and we will have these ponds in which water systems and other things go. So the premises of it, of course, being that building a new campus creates CO2 emissions, but in the large run, I do feel like it's more sustainable, and also for the community, because we need a center for resilience, in my opinion.
Thank you.
Hi, thank you for the wonderful presentation. I'm from Riga Students University. I have not so much maybe question as, I don't know, a comment or suggestion, because I'm part of the project dealing with like basically investigating sustainability and green transition in Latvian state hospitals, which are also really large institutions facing a lot of challenges. And so it's actually really great to see how like there are these small kindergartens that you manage to get this green flag. It's like, you know, it's a very different scale to actually reach that. But one observation that I have is like, even though I'm kind of in this topic as well, it's the first time I hear about all your achievements in this regard, and that's also something that we see in the healthcare, that hospitals, there's some doing really great things and the others just know nothing about it. Like there's this kind of lack of information and experience exchange in Latvia among institutions, where we could really learn a lot from each other and get inspired by each other perhaps. So yeah, that's really less of a question and more of a maybe a comment or suggestion. But thank you. It's I'm happy to see how much Latvian University is doing.
Thank you. If I may quickly comment on that also, I think half a year ago we started a non-official group of sustainability experts from Riga Technical University, Riga Students University, and from the University of Latvia. And we are working towards a common program for educating students and staff on sustainability. So finally, finally, we will be collaborating in this matter.
Thank you, Renard. It was really impressive to hear what you have achieved during since establishment of this council. But in fact, I believe there is a lot of other prospects for further development, and one of them is quite formal. I don't know if you have an option to participate or to tell about your activities and plans within the course which is mandatory for all the university students, which is environmental protection. So it would be a very good option or instrument to promote your activities and to make all students familiar, not only those which are living in campus, but also those who will come and I believe will enjoy the possibilities of environmentally friendly and at the same time technologically advanced environment for development, their future careers and lives. Thank you, and my best wishes for further development. Thank you.
Well, thank you for the continuous support and a really great idea will be noted.
Thank you, Renat, for the presentation. I was wondering, do you have any suggestions for researchers in terms of sustainable research?
Well, what I can kindly ask is that researchers are invited to do research at our urban meadows, botanical gardens, around the community that has formed around it. But in terms of advice for research to be greener, that is a tough one. But on this topic, we will also have a blended intensive program together with universities from Ireland and Poland. And we will be teaching researchers how to make their research a bit greener. So I will keep you updated with the results that come out from that.
Thank you. I actually have one comment on this. One way to make your research more sustainable is to make your data open. Like when you do it, when you make them open, when you describe them well, when you make your methods or explain your methods well, that means someone else doesn't have to repeat what you already did. So that's also towards sustainability. Thank you.
Thank you. Okay, thank you.